The last three factors that I would like to discuss that are produced by the stomach are pepsin, mucus, and intrinsic factor. Now, as we saw in the salivary secretions, that the acinar cells will produce amylase, which is an enzyme required for digestion of starch into glucose monomers. So that is the beginning of carbohydrate digestion. The beginnings of protein digestion will begin in the stomach. And we have a type of cell called a chief cell, which is located in the cryptic pits in the stomach infolds. And the chief cells are going to be producing a protein called pepsinogen. Pepsinogen is a precursor to the general classes of a protease. So these enzymes are responsible for digesting proteins into small peptides and amino acids. Recall that we have different classes. We have exoproteases and endoproteases. These digest from the ends of proteins. Pepsinogen endoproteases will digest in the middle. Pepsinogen will digest in the center, so it's an endoprotease. Now, the important feature here to remember is the suffix ogen. Ogen is inactive. And this makes sense because when the chief cells are making pepsinogen, you don't want an active protease inside your cell to digest your own proteins. So the cell will make the inactive form of the protease. The activation of pepsinogen occurs in the lumen of the stomach. And this is requiring low pH. This is what's called an autoproteolysis, that there's a conformational change in the pepsinogen molecule so that it is able to essentially cleave its tail, hydrolyze its tail off, releasing a small portion of the protein, and then the protein becomes the active protease. Then the pepsin, its optimal activity is at pH 2 to 3. So its optimal enzymatic activity is in low pH. So it's digesting the protein in the diet. Now what happens when that pepsin exits the stomach? Into the duodenum. Now we'll talk about that, how the duodenum can adjust the pH and raise the pH to actually approaching 8. So now remember, enzyme activity has an optimal pH. So if we were measuring enzyme activity and we had a scale of pH, let's say this is 2, this is 10, a pH optimum means that the enzyme activity is high in a certain pH range. When we're outside of that pH range, the enzyme activity goes to either close to 0 or 0. In this manner, the pepsin is active only in the low pH environment of the stomach. Now, the next cell type are called goblet cells. And I'm sure you've seen these in histological examinations, a set of tissues. Goblet cells are very unique in that they are filled with a translucent material. And this translucent material is a mucopolysaccharide. What is a mucopolysaccharide? These structures tend to have very short protein domains and very long carbohydrate domains. This gives them their mucus-like properties because the long carbohydrate chains on these proteins makes these very viscous because they bind a lot of water, and they also interact with lots of hydrogen bonding together, and so they form a sticky, gelatinous substance. Now this is important on the stomach because the goblet cells are located all along the stomach lining, and they are secreting this gelatinous layer which will sit on top of the cells. So if we have a series of cells like this, goblet cells will be intermixed in these, and it will form essentially a gelatinous layer over the cells, separating the top of the cells from the lumen. So what is this doing? Well, first of all, remember that we have active pepsin in the lumen. That pepsin, if it can access the top of the cells, it could digest proteins and damage the cells. So this gelatinous material prevents the pepsin from attacking and digesting hydrolyzing proteins at the cell surface. The other important point is that the cells are secreting bicarbonate ion. These are the goblet cells secreting this. Now remember, bicarbonate ion is basic. So if we have low pH in the lumen, lots of protons, the bicarbonate ion will associate with these protons to neutralize them. This gelatinous material, matrix on here, will tend to hold that bicarb in through ionic and hydrogen interactions, hold that ion close, and so any protons from the low pH that get into this, diffuse through this gelatinous matrix, will be neutralized by the presence of the bicarb. So the pH at the cell surface of the stomach lining is 7, neutrality. And therefore, the acid is not contacting the cell surface and not potentially disrupting or denaturing any important vital proteins on the cells. Now the final secretion product is actually coming from the parietal cells that are making the acid, secreting acid. These cell types are secreting intrinsic factor. Now what is intrinsic factor? Well, one of our essential vitamins that we need is vitamin B12. And we'll talk about vitamin B12 later in the block when we start discussing anemias. Vitamin B12 is critical for 1-carbon metabolism, and it's also critical for maintaining red blood cell in an antioxidized state. And getting vitamin B12 in our diet is very important, but how do we do it? Vitamin B12 is very, very hydrophobic. So unlike our hydrophobic fat-soluble vitamins, Vitamin B12 binds to proteins very tightly in our diet. And this is how they are accompanied in our diet. The vitamin B12 must be accessed. In order to do that, the protein portion, and this is a non-specific interaction, it just binds to any protein. We need pepsin degradation. We need low pH denaturation of the protein for the pepsin to get in there and fully hydrolyze. And that is going to release the vitamin B12. Now recall, B12 is very hydrophobic, so it needs a carrier. That carrier is intrinsic factor. This complex of intrinsic factor and vitamin B12 will travel through the intestines to the ileum. There are receptors present on the epithelial cells of the ileum that will internalize the intrinsic factor B12 complex The intrinsic factor inside is degraded, releasing the B12. The B12 enters the bloodstream, secreted from the basal surface of the epithelium, and there will complex with transcobalamin. Transcobalamin is a plasma protein that specifically binds B12. The transcobalamin B12 complex can then enter cells as needed.